Today we will discuss about lac operon in E. coli. Lac means lactose. Lactose operon in E. coli. In 1961, Jacob and Monat two microbiologists proposed the Operon model for the regulation of gene in E. coli. Operon is a part of genetic material. Operon is a part of genetic material. That is, operon is a part of DNA. Operon is a part of DNA. In broad sense, operon is a functioning unit of DNA. Operon is a functioning unit of DNA containing cluster of genes under the control of a single promoter. Okay. Operon is a functioning unit of DNA containing cluster of genes under the control of a single promoter. Operon are of two types. One is inducible and second is repressible. Now I will draw the lactose operon. This is the lactose operon. Here P represents promoter. P represents promoter. O represents operator. Lac Z, Y and A these are structural genes. These are structural genes. I represents a regulatory gene. I represents regulatory gene. Which coat a repressor. Okay. I regulatory gene which coat repressor. Okay. Now what is exactly happen? Lac operon is for metabolize the lactose, that is breakdown of lactose. Lac operon is for breakdown of lactose. Okay. Now we already known that operon is a functioning unit of DNA. This is the DNA. Okay. Functioning unit of DNA. This is the functioning unit of DNA containing cluster of genes. Cluster like Z, like OY, like A structural genes and operator promoter, and under the control of a single promoter. This is the single promoter. Okay. Operon is a functioning unit of DNA containing cluster of genes under the control of a single promoter. Now what happened? RNA polymerase bind to the promoter. RNA polymerase bind to the promoter, and these genes are transcribed. Okay. And transcription, then number of proteins we found. Now what is actually happen in Operon in E. coli? I here the regulatory gene I, which code the repressor. This is the repressor RNA. It makes repressor proteins. Okay. I regulatory gene. This is transcribed repressor RNA and it produces repressor protein. Then what happened? This repressor protein bind to promoter region. Okay. This repressor protein bind to promoter region. Then what happened? RNA polymerase cannot bind to the promoter region, so transcription is not possible. In general, transcription here not possible because of repressor protein. Repressor RNA transcribed and repressor protein is produced which bind to the promoter site, and structural genes are not transcribed and transcription is stopped. Actually lac operon is a switch off operon, switch off. Then when in media lactose is present, then what happened? When in media lactose is present, here the lactose. Repressor protein, and lactose bind. Here when in media lactose is present, lactose bind to the repressor protein. Then not actually lactose, it allolactose. It bind to the repressor protein. then promoter region is free. Promoter region is free. Then what happen? The RNA polymerase bind to the promoter region and lac ZYA transcribed. Transcription. Three types of protein are translated. Beta galactosidase, permease, and transacetylase. When lactose is present in media, then the operon is switch on and the structural genes are transcribed. Ultimately, beta galactosidase, permease and transacetylase enzymes are produced. Beta galactosidase is primarily responsible for hydrolysis of the disaccharide, that is lactose, and transacetylase catalyze the transfer of an acetyl group from one molecule to another. In other words, permease, it increases the permeability of the cell to beta galactosidase. Okay. So, in media, lactose present, operon on, lactose absent or present in low amount, operon off. Because the lac operon is for the breakdown of lactose. Okay. When in media lactose is absent, there is no needed of lactose operon. So when lactose present in media, operon is on, and lactose absent in media, operon is off. Thank you.